Okay, so we are officially recording again. Gonna move stuff over so I can keep an eye out on the chat. Okay, so let's get back to where we left off here. Is there anyone who cannot see my screen? Okay, great. I'll assume you guys can all see it. Okay, so we now have our samples of interest, in our case, flight and ground control RNA extracted from the soleus muscle of mice. And our first step is going to be to isolate RNA. Once we do isolate that RNA, there's only going to be a certain portion of it that we're going to be interested in, depending on what we want to look at. So if you isolate total RNA from a sample, the majority, I know it's kind of on here already, but does anyone know what the most abundant RNA in our cells is? You guys could type it in the chat or shout it at me. Yeah, our RNA. So ribosomal RNA is all over the place, which makes sense because proteins make us do everything we do. So there's a lot of ribosomal RNA. And so ribosomal RNA is fairly well conserved, not horribly interesting. And so the fact that it makes up over 90% of all the RNA in our cells means that it would take over all of our sequencing. And so all that we would ever sequence is ribosomal RNA, which again, not very interesting. And so we're going to want to remove or at least reduce drastically the amount of ribosomal RNA that we have in our samples before we do go any further in the sequencing process. And so there's a couple different ways you can reduce or remove ribosomal RNA. And so just to kind of remind you guys, I know we talked you through this, but pictures help. So if we're looking at message RNA, for instance, and so usually when you're looking at differential gene expression, message RNA is going to be one of the things that you're really going to want to take a look at. And so I'm going to use this here as an example. So we have our pre-message RNA equipped, so we see the 5' and 3' ends here, and then we see our exons colored in red, our introns colored in light bluish gray. And what happens as pre-mRNA is matured into message RNA as we see splicing happen. So those introns are removed and the exons come together. And then we also see a 5' cap added on at the 5' end. And then we see a poly A tail added onto the 3' end. So hopefully this is bringing back some molecular biology for you guys. And what we can do to help clean up or remove ribosomal RNA is, one way is through positive selection by taking advantage of this poly A tail. So we can create oligos that are complementary to long strands of A's or long strands of T's, depending on which forward or direction mRNA you have. And then we can use that to bind to the poly A tail and pull out the message RNA. So this is a way to enrich for message RNA in your samples, because then we would only carry that pulled out mRNA through the rest of the process. However, although mRNA is very interesting, especially for differential gene expression, which is what we're going to be doing, when you do that, you'll also kind of minimize the other types of RNAs that are there. So as some of you guys may have learned from some of your courses, there are a bunch of really cool RNAs that do important stuff, such as microRNAs. We also have long non-coding RNAs, there's SNO RNAs, there's small nuclear RNAs, there's all sorts of RNAs that might be interesting to us. And so we might not want to just look at message RNA, but we might also want to look for differential expression of or changes in the amounts of microRNAs or long non-coding RNAs or some of these other RNAs that are not message RNA. And so the other way to do it is, rather than to positively select for mRNA by pulling down the poly A tail, we can also instead create oligos that are complementary to ribosomal RNA and remove the ribosomal RNA from the sample. So what does your relative percentage of the different types of RNAs look like when you do both those approaches? So this is an example here. This is a human RNA, universal human RNA reference that this was done on. And so we go from over 91% of our total RNA being ribosomal RNA to when we do a poly-A, or also known as mRNA enriched selection. So this is where we're taking advantage of that poly-A, and we are only pulling out RNA that have a poly-A segment. And so what we see is a huge enrichment in the amount of exonic RNA that we're capturing. And we still do also capture some intronic and intergenic RNA as well. Any guesses as to why we would also capture some other types of RNA, not just mRNA? You guys either write it in the chat or unmute your mics. No guesses? So as I mentioned, when we do this positive selection to look for that poly-A tail, we create an oligo that's complementary to a long strand of A's. If those regions happen to have a lot of A's by chance, absolutely, yes, Aylin. That's exactly what I was looking for. So poly-A tails of message RNAs are not the only location necessarily within our transcriptome where we find long strands of A's. And so any other region that has long strands of A's will also, of course, bind to the oligos and be pulled down along with that message RNA. And what you can see here is when we do enrich for message RNA, you see how much reduced the ribosomal RNA portion becomes, which is exactly what we're looking for. But again, we have the caveat of, although we still have captured some other types of RNA, we did that in a biased way, right? Because those are going to be ones that have long strands of A's in there. And so rather than select specifically for poly-A tails, instead, as I mentioned, we can remove ribosomal RNA. And so this is an example of what happens to the percent of RNA as you do that. And so what you can see here is now you've enriched all the other types of RNA and still drastically reduced your ribosomal RNA. And so in GeneLab, because we want to make as much data available, Lauren, are you on?
Yes, I'm here.
Okay, just heads up to keep an eye out on the participant list just in case people are in the lobby.
Oh, I'm sorry. Can you make me a co-host again?
Oh, that's my fault. Yeah, let me do that.
No, I should have reminded you.
Okay, you should be a co-host now.
I am. Thank you.
Okay. Sorry about that, guys. Let me go back to sharing my screen. Okay. So as I was mentioning, pull out the chat again. Okay. As I was mentioning, oops, let me go back one more. In GeneLab, because we want to make as much data available to our users as possible, we decided that the standard that we use when we sequence samples in-house is we use the Ribo Depletion method. So we remove ribosomal RNA so that way we still maintain the levels of other non-mRNAs in our sample so we can capture those in our sequencing. Okay, so now that we know a couple different ways to enrich for what we would be interested in sequencing in the context of RNA, let's talk about how we prepare libraries and get these samples ready for sequencing. So we're going to be looking specifically at the Illumina sequencing workflow. It's because Illumina is an incredibly successful biotech company that specializes in next generation technology, utilizing that pyrosequencing method or sequencing by synthesis that we reviewed before the break. So about 90% of all sequencing worldwide is performed on an Illumina instrument, including here at GeneLab. And as nanopore sequencing technologies continue to increase in popularity and become less and less error prone, we're actually seeing an increase in that. So I anticipate that Illumina's reign on the sequencing world is going to slowly be reduced. However, they still are the worldwide leader in sequencing. and their technology is amazing. So the Illumina sequencing workflow has 4 major steps in it. So the first one is constructing the library, or library preparation, then cluster formation, followed by sequencing, and then lastly, of course, data processing and analysis. So we're going to walk through in painstaking detail all of these steps. So in step 1 depicted here, and all this stuff is pulled from the Illumina TrueSeq Stranded Total RNA Reference Guide, if you guys want more information. So in figure 1 here, our first step in library preparation for our context and how we do it at GeneLab is we're going to do that Ribo Depletion step. So we're going to remove ribosomal RNA from the total RNA that we extracted from our samples. And how we do that using the Illumina Ribo0 kit is this kit is equipped with oligos that are complementary to ribosomal RNA. And these little oligos also are biotinylated, which means that they're magnetic. So we can actually pull them out with a magnet. So what we do is we take our total RNA, we add in those biotinylated oligos that are complementary to ribosomal RNA. Those will then bind to ribosomal RNA in our samples, and then we use that magnet to pull out that ribosomal RNA from our samples. So after that, we should be left with ribosomal depleted total RNA. But transcripts can be very long, and as I mentioned previously, Illumina is a method of sequencing short strands of RNA. And so we want to chop up our RNA to make sure that the strands are short enough to be able to be sequenced on the Illumina platform. And so there's an enzyme that we added in, or it's actually not an enzyme, sorry. It's a metal, actually, to reduce the bias when it's randomly chopping up your RNA. And so we'll add that ion in and chop up our RNA into small pieces. And the size of the fragments that you want will depend on how long you want to sequence. So at GeneLab, we sequence at 150 base pairs read length, and we sequence in both the forward and reverse direction. So our goal is to get RNA fragment sizes at roughly 250 to 300 base pairs is what we're aiming for. And how you can change the size is by doing more or less digestion. So the longer you digest your RNA, the smaller your fragments are going to be. The shorter you digest your RNA, the longer your fragments are going to be. And so you really want to hit that sweet spot. And then we also perform filtering steps, so that way we size select and make sure that we're selecting for the size of RNA fragments that we want to sequence. Okay, now that we removed our ribosomal RNA and chopped up the remaining RNA that is there, our next step is to turn that into DNA, because we only sequence DNA, not RNA. So similar to the reverse transcriptase step of our RT-PCR that we talked about is taken advantage of in the Illumina library preparation protocol. And so we synthesize the first strand of cDNA from that RNA template. And then after we create that first strand of cDNA, we then create the second strand of cDNA. And Illumina kits are trying to make their DNA stranded, so that way when we're doing our sequencing, we're always sequencing the same strand. Because remember, this comes from double-stranded, so this helps control and regulate when we go and do our quantification if we're always looking at the same strand. And so in order to make our library stranded after creating that first cDNA molecule from the RNA template, when it does the second strand synthesis, instead of adding in dinucleotide, so instead of adding in T's basically and their DNTPs, it adds in U's. So basically it makes a complementary RNA strand. And so that's what you can see in blue here. So you notice that we have U's instead of T's, and we'll see how that allows it to be strandedness later on in the library preparation. So as right now we synthesize our first strand of cDNA, we then made complement or synthesized the second strand of cDNA, but made it with U's instead of T's. And then our next step is to add to polyadenylate the 3' ends of our cDNA. And the reason that we do this is because, do you remember previously when I was giving the overview, I said that we have to add stuff to our target sequence to make sure that it latches onto the flow cell? How the Illumina library preparation method works is it utilizes these A's that we add onto the 3' end. When we add on those adapters, it'll have a complementary T, and we can use an enzyme called ligase that'll actually glue these complements together. So after we add in these adenylated, these A's to the 3' ends of our cDNA insert, we're then able to add in those adapters. And so this is what the adapters look like. So we have a P5 and a P7 region. So these regions are oligos that are going to be complementary to the flow cell. So that way it's going to be able to latch on to the flow cell during the sequencing process. Or I should say just prior to sequencing. And in addition to the P5 and P7, we also want to add in an index. Anyone have any guesses as to why we're adding on an index? You could type it in the chat or shout it.
Sorry, what's P5 and P7 again?
So they're just little short oligonucleotides. So they're basically A, T, C's, and G's that are going to be complementary to the flow cell. So the flow cell is where the sequencing happens. So in order to make sure that we get the piece of RNA, which is now cDNA, that we want to sequence attached to the flow cell, we have to make sure that it's part of it is going to be complementary to the flow cell.
Ah, got it. Thank you.
Make sense?
Yep.
And we're going to watch an awesome video that's really going to hopefully bring this all together. So yeah, primer attachment, keep track of orientation of the strand, all great guesses. So we actually add on an index so that we can keep track of which pieces of RNA came from which sample. So when we do the sequencing, we sequence a lot all at once. And so we actually combine samples. So we'll combine all the samples from our flight group and all the samples from our ground control group into one what's known as a pool. And then we sequence it all together. But because we mix it all first, we need to find a way to figure out which sequences came from which samples. And so before we mix them all together, when we're doing this library preparation step, we give each sample its own unique index. And again, these index is just another chunk of A, T, C's, and G's. It's just another short oligo, and it's going to be unique for each sample. And in this adapter here, this is known as single index because there's only an index on one end. You'll notice there's no index associated on the P5 side of the adapter. We'll take a look at what that looks like though. And so this is single index here. In addition to this P5 and P7, which again allows you to attach the region that you want to sequence onto a flow cell, and then also having this index here that allows us to keep track of which RNA came from which sample. We also have to add in some primers. Anybody know why we would have to add in primers? Yeah, so those are both pretty much right on track. PCR and some more can be built. So remember, we sequence by using sequencing by synthesis, right? So sequencing by synthesis, similar to what happens in a PCR reaction, we're creating a copy. And so in order to create the copy, the first thing we have to do is we have to have an annealing of primers to the region that we want to sequence. And so we have to have a portion on this adapter that's going to be complementary to the primers, so that way the primers can hop on, which will allow the polymerase to hop on and start building the strand that's ultimately going to be our sequencing by synthesis. And so it's kind of, again, just like a remarkable way to bring together all of these technologies that have come before it. And then this T, of course, is added on, as I just mentioned. Anybody remember why we would have a T here? So take a look at where we're at with our target binds to A. Yeah, exactly. That's right, Hannah Lee. So what happens is this causes what's known as an overhang. So I don't know if any of you guys have done any cloning, but this is a technique that's used all the time in cloning with something called restrictive enzymes, which are a lot of fun to work with. But what this does is it provides a little overhang here. So we see that this extends out just one more base pair, and that base pair is complementary to the A that we just added on in the last step to our DNA of interest. We'll see DNA of interest. And so now when we add these adapters along with our cDNA here, and we also add in the enzyme ligase, ligase will help glue those A and the T together, giving us what looks like here. So this will be our region of interest that we want to sequence, and now we have an adapter latched onto it. Adapters can be loose adapters that aren't bound to your region of interest are a nightmare in sequencing. It actually causes a whole mess of problems. So right now at this step, after we add on the adapters, it undergoes a filtering step to size select, so that way you remove any of these kind of adapters that are still floating around in your solution. And so as I mentioned, this is considered a single index library because there's only an index on one strand here, the strand associated with P7. This, of course, will help us. What's known as demultiplexing is when we identify the index and we know which index we added to which samples. So then we're able to use that information to say, okay, this sequence belongs to this sample, this sequence belongs to this sample when we're doing our data analysis. However, there are some little tricks and weird things that can kind of go on during sequencing and cluster formation specifically before sequencing of where these indexes sometimes will hop. So they'll actually hop around and become associated with another cDNA, which could have possibly come from another sample. And so it's always better if you can have two indices instead of just one. So instead of just an index associated with the P7 side of things, it's also good to have an index associated with the P5 side of things. And that's called having dual indices. So these are now unique dual indices. So now when we go to assign this back to a sample, that sample will have to match the I5 index as well as the I7 index that we gave for that sample. So it just increases our confidence that we're assigning the sequences back to the right samples during the data processing. And then I also want to point out just another type of index, which is actually the one that we use in GeneLab. And we'll kind of get into this a little bit more in depth later, is that during the next step that I'm going to go over in processing is the amplification step. And during the amplification step, we can cause artificial duplications of sequences of interest. And so we want to be able to detect those, since those would have been added artificially. And in order to do that, we at this step can use an index that also contains a unique molecular identifier. And this is going to be a random set of base pairs that you're going to add on that'll help you identify technical duplicates later on in your analysis. And I'll get to that later. And some of you who are going to be doing internships with us, although none of you guys put this as your number one project choice, but hopefully we can change some of your minds into being able to come up with a pipeline to be able to identify and remove duplicates based on those UMIs. And again, I'll get into a little bit more about that later. And so the next and just about the final step of the library preparation is to enrich the DNA fragments. So now we have our cDNA. It's currently double-stranded. And remember, when we added on that second strand, we added on U's instead of T's. And the reason we did this is to have, I got a question from Leah. During PCR, there's a risk of generating primer dimers. Is that still a risk here? If so, how do you deal with that? Yeah, primer dimers is a huge issue in the PCR step. So remember, after we create our libraries here, after we add on our adapters, at this step, we do a size selection. So we try to remove anything that's small and hopefully remove our primer dimers. But you're right, if some of them kind of slip through the cracks, which can happen for sure, and has happened to us, they will get amplified during that PCR step for sure, because that PCR step is utilizing these this priming region right here. So yeah, good point. And so that PCR step, if we have adapters in there, it will definitely amplify those adapters and adapter dimers, which is a big no-no. So during that PCR step, so what we're doing now is after we created our double-stranded cDNA from our samples, we want to amplify it just to make sure that we have enough of our sample to be able to sequence. And so we go through a PCR step here, and we do it at a very low number of cycles because we don't want to blow up and create a whole bunch of artificial duplicates. So we do a low number of cycles here, and during this step, the polymerase that we use, it isn't able to incorporate any new nucleotides if it runs into a U. So this is how the trick that Illumina uses to be able to make their library stranded. So everything we sequence is from the same strand. And so as you can see here, during this amplification step, the first strand of the cDNA synthesis process that we synthesize, that'll go ahead and the polymerase will move right forward, and then that's the strand that's going to be amplified. So basically only this strand that's in this direction is going to be amplified during this step, because when the polymerase goes to try to amplify its complement, as soon as it hits its first U, it's going to stop. And so it won't amplify that strand. So that'll give us nice stranded libraries. And then of course, after our PCR step, we then go through another final cleanup. And again, remove anything that might be too small in hopes of removing any, of course, anything that we added in here. So loose dinucleotide triphosphates, loose primers that we use during the amplification step, and as Leah pointed out, any adapter dimers that may still be in here. And then after that filtering step, we have our final stranded libraries. And again, this one here is depicted as a single index. So you only see that one index in blue here on the P7 side. We don't have any on the other side. In reality, in GeneLab, we always have unique dual indices as well as that UMI. But again, we ingest data from all experiments. So some experiments we have samples that have single indices, we have samples that have unique dual indices, we have samples with and without UMIs. So we really need to understand how the libraries were prepared in order for us as bioinformaticians to know how to process that data. Any questions about any step of the library preparation before we get into evaluating these libraries? What exactly is a duplicate? Great question. So remember, when we are doing the sequencing, ultimately, we want to know how much starting material of each gene, if we're looking at gene expression, which is what we're going to, we want to know how much of each starting material was in our original samples, right? And so we want to make sure that we are not artificially increasing or adding to the number of copies of any gene. And so, of course, you run that risk when you do this PCR step because you are purposefully amplifying the stuff that's there. And so if we had a gene that was really high abundance, so there was a lot of copies of a particular gene, when we go through and do this amplification step, those genes that might have been more highly expressed, so the transcripts that are more abundant, have a higher likelihood of, when we do this PCR step, they will be duplicated, and maybe one of those artificial duplicates is going to be what's sequenced. And so now we will have artificially added an extra sequence that we're going to then quantify and say, oh, this has, +1 sequence, when that wasn't the case biologically speaking. So that's what I mean by a duplicate, and specifically a technical duplicate. Does that answer your question, Rashi? Okay, great. And we're going to talk a little bit more about duplicates a little bit later during the analysis or evaluation of our sequencing data. So we'll revisit that concept. Any other questions about library preparation before we move on to evaluating our libraries? Okay, So our next step, now that we have our libraries prepared, is to check the quality of our libraries. So this, again, is an incredibly important step, and it's kind of technical in how we do it. So I know that we kind of had a little debate about whether or not to show it, but it's just so important and will help us analyze our data and troubleshoot when things go awry. So I'm going to go into a little bit of detail about how we do that. And we evaluate our libraries using electrophoresis and electropherograms. Does anyone know what an electropherogram or what an electrophoresis is? Christina, are you talking? If you are, you're muted, so unmute yourself.
Sorry, I hit the
No
video
problem.
instead of the mute. Electrophoresis allows you to separate out sets of DNA that you have based on size.
Yes, absolutely. So kind of what a nice big size gel looks like is here. And so we have a polyacrylamide gel usually, and it's sitting in this buffer of liquid. And what we do is we load in our samples in the different wells here. And then we, in addition to our samples, we usually also have a marker. And that marker is going to have DNA of known sizes. And then we actually color code the DNA of known sizes so that each one of these bands here will be a different color. And then based on that color, we'll know what size it is. And that's how we can kind of get a good guess of what the size of the DNA in our samples is. And so once we load our samples into the wells of our gel and we have this buffer, we then apply a current. And so we apply our current from negative to positive. So negative being where the samples are loaded, positive being where it's going to run to. Any idea why it's in that direction?
Because DNA is negatively charged. So
Absolutely.
It runs positively charged.
Yes. So DNA has phosphogroups on it, which are negatively charged, and so they will be attracted to the positive electrode. If you ever hook up your electrodes in the wrong orientation, you will lose your sample. I have done that more than once, and it's really frustrating. So you always want to make sure you put your current in the right direction so that your DNA is flowing from negative to positive. And then what flows faster? Smaller DNA, smaller fragments of DNA or larger fragments of DNA? Which ones are gonna travel faster? Sorry, go ahead.
Smaller fragments.
Yeah, so your smaller fragments are going to have an easier time making its way through the polyacrylamide gel, and so they're going to travel faster. So your smaller, lighter stuff is going to end up on the bottom, and your heavier DNA fragments are going to end up on top. And then what you can do, and there are computers that kind of do this in really small gels, and they generate what's known as electropherogram. So electropherogram is a way to read the information generated through electrophoresis. And so what happens here, so in this instance, as I mentioned, you can add a marker. And so your marker will be of known size. And so then you'll be able to tell based on the location of your sample of interest what size your sample of interest is. Another way to be able to distinguish is how quickly your samples are identified or how quickly they make peaks. So the sooner a peak comes off, that means it traveled faster. And so that's going to be a smaller size. And so you could, in electropherograms, you'll either see the x-axis written out in time or written out in concentration, whether it be an actual concentration like a micromolar or something like that, or in base pairs, which is what we actually see for library electropherograms. And so just kind of going over what this looks like. So in this case, we have fluorescence here. So in this case, the DNA was labeled with fluorophore, and so fluorescence was being measured to identify when DNA is coming off of the gel. And we have our marker here to indicate that we should start looking for peaks right after that marker. And then again, if you're looking at just RNA and running it through, we're going to see like gas chromatograph, but for DNA, exactly. So this is more like liquid chromatograph, but a little older school. Gas chromatograph is a little bit fancier. And so here, as these are coming off, of course, if we have an RNA sample, if we did not clean it up or remove ribosomal RNA, that's going to be the biggest things we see, right? Because we just saw that over 90% of the RNA in our cells are ribosomal RNA. And so we know that there's 3 distinct ribosomal RNA fragments. We have this really small 5S fragment, followed by the larger 18S fragment, and then the largest 28S fragment. And so we'll see these nice, very large, very distinguishable RNA or ribosomal RNA peaks when we look at an RNAsepherogram of just total RNA that hasn't been cleaned up or filtered. And so, of course, when we're looking at our library preparation, do we want to see any of these specific peaks in there? No, right, Leah, absolutely not. The goal is to remove as much ribosomal RNA as possible. And so this is what an electropherogram looks like from an RNA library preparation. And so what you can see on the right here is what that gel looks like. And then on the left is the electropherogram representing this gel. And so as you can see here, we have a heavy marker, which is shown here in purple and seen here on the electropherogram. And as you can see here that I mentioned, this x-axis is in base pairs now. And again, we know what the base pairs are because we know the size of these markers. And we can then deduce the size of our libraries based on the size of those markers. So that's how that's calculated. And so we have this heavy marker over here, and then we have this nice big peak, which is our library right here. You'll notice that we don't really see kind of these 3 or mostly these 2 peaks anywhere in here, which is great. So that shows that we don't really have RNA or ribosomal RNA contamination. However, here's our low end marker in green here. And then again, we have our high end marker in purple or blue here. And then we have our library, this nice smear shown on the gel is shown as this peak here. But then we have this other peak. You can see this band here, and then you can see it right here very clearly on the electropherogram. What the heck is that? Any guesses? Remember, this is a library that we made during that library preparation we just went over. Any guesses as to what this peak could be right here at about 150 base pairs? Index, yes. These are the dreaded adapter dimers. So adapter, I think I mentioned the size of adapters. But yeah, Hanley, you're absolutely right. So those adapters are roughly 75 base pairs in length in total. Again, that has some plus or minus depending on if you have dual indices or just one index and whether or not you have a UMI or not. And so what can happen is those primers or those adapters, they can bind to each other and basically double their length. So if the end of that adapter, let's go back with the adapter. So basically this adapter can latch onto this, in which case you would have, I guess I do have a picture of this in the next slide. So you would have your P5 side and then your P7 side making what's known as an adapter dimer. So then you have roughly 75 base pairs of this side, roughly 75 base pairs of your P7 side to give you roughly 150 base pairs in total, which you will see if you have that contamination and did not properly filter out for adapter dimers. So this is not a good library. I would not sequence this. So libraries, rather than evaluating one at a time, we can actually evaluate multiple libraries at a time, and we can run really teeny tiny gels that are hooked up to computers to generate electropherograms automatically, which is really convenient. And those machines are called bioanalyzers or tape stations. And that's what we use routinely to assess the quality of the libraries that we prepare, and so does all sequencing facilities. And so here's some examples of some good libraries. So again, I have the electropherogram on the left, and I have the respective gel on the right. And as you can see in both of these, we see our lower end marker, we see our heavier marker, we see a nice strong peak for our sample library, and then we see a very small, if anything, around the adapter dimer size. So these would represent pretty good libraries that I would go ahead and move forward sequencing. This is a really bad library. So in this library here, we have again our small marker over here on the left, our heavier marker over here on the right. We have just a little bit of this containing our actual sample library, and we have a whole lot of adapter dimers. So this library is trash. I would throw it out and start again. I wouldn't even try filtering this. If you have just like a smaller peak, you can go through an extra filtration step to see if you can kind of remove those adapter dimers. But every time you go through an extra filtering step, you're reducing the amount of sample that you have, and you still need to make sure you have enough sample to sequence. And so just to kind of summarize what I've been talking about, when assessing library quality, you want to look for and make sure that you do not have adapter dimers. Why is that? We're gonna actually get into more of this, I believe, in the next slide. But any guesses why adapter dimers are such a problem? It's noise in the data. That's a good guess and pretty accurate. Any other guesses?
Actually, I had a question about that. I mean, if you have low levels, can't you just remove it with code? Like, you know the sequence, so can't you just say remove this sequence wherever it appears?
You absolutely can do that. However, the problem is that the flow cell in Illumina, it's actually biased towards smaller fragments. And so what happens is that your adapter dimers are going to be smaller than the proper libraries that contain your region of interest that you want to sequence. And so when you load your sample onto the flow cell, the majority of those little oligos that are on the flow cell that you're attaching your sample of interest to will be taken up by the adapter dimers so much more quickly than the region that you want to sequence. So much so that the majority of the sequence you're going to get back, even with a little bit of adapter dimer contamination, will be a lot of the sequence. And so you're basically throwing away money because all you're doing is sequencing adapters and not your region of interest. But you're absolutely right that you can indeed remove that in silico, for sure. And I'm going to show you a little bit about just how little contamination can lead to a whole lot of problems in terms of what's sequenced. The other thing that you want to check for, which I haven't really mentioned yet, is library size. And so the goal here is to make sure that your library size is consistent with the desired sequencing cycles. So if you guys remember, I mentioned this just once previously, but if you have, if you're sequencing at 150 base pairs, sequencing length, and you're doing it in paired ends, so in the forward and the reverse direction, what's an appropriate size of the region that you have, the fragment that you want to sequence? So 150 base pair length, paired end sequencing, so sequencing in both directions. How big would you want your insert? Any guesses? Yeah, right around 300 or just under. So it's really nice when you're sequencing in both directions. You want to be able to fully sequence your fragment. So if you were to have inserts of exactly 300, you would sequence 150 in the forward direction and 150 in the reverse direction, and you would cover your whole sequence, but it's nice and comforting and helps a lot with the alignment if there is some overlap between your forward and your reverse read. So usually about 20 base pair overlap to 30 is pretty ideal. And so what I would look for at paired end 150 sequencing is I would want to see those inserts right around 250 base pairs. But then remember, you also have your adapters that are attached onto either side of your insert. And each adapter is about 75 base pairs. So you're going to add in about 150 base pairs to that. So you have 150 base pairs total making up your adapters, plus your roughly 250 base pairs of the region of interest that you want to sequence. So you're looking at a library size of right around 400 base pairs. It's kind of what you're shooting for if you're sequencing with the parameters that I just mentioned. Of course, some people sequence with less cycles. Really, the high end of Illumina sequencing, I think it's 500 cycles total, so 250 by 250. But we usually just use that for metagenomics sequencing. For RNA sequencing, our standard is 150 paired end in either direction. And so we want to see around 400 base pairs. What you see here is that these libraries are right around, I guess I kind of have this here too. Is the amount of smearing of the library band significant? Like, is it better to have a cleaner, distinct band, like in Good Library 1 versus a smear band in Library 2? Great question. So this is so when you do your chopping at that first step of library prep, your goal is to get roughly everything relatively the same size. So the tighter that peak is, the more uniform your insert size is, which is nice because that minimizes bias when you go to run it on your flow cell. So if you have a broader range of size, ultimately of your inserts, right, because they're all getting the same size adapters ligated onto them, what you'll have is it'll tend to be biased when you do your sequencing, is that the smaller fragments will be more likely to be sequenced than some of these larger fragments. So yeah, if you can get that a little bit tighter, that's definitely better, but you're always going to have some range. So especially because of the filtering we do, there's about, I don't know if Marie's on, but I think there's around 100 to 200 range in the pores for the size filtering. Is Marie on? Marie's part of the sample processing lab in GeneLab. She actually had a lab day today. Great question. And so I know we already talked about that. I gave the example here as Paradigm 100, but as I talked about it, I said Paradigm 150. So just asking what the range of library 1 is. So what do you guys guess the range of library 1 is here? Any guesses? You got the base pairs down here. So see the range is probably from around here to around here. Yeah, 150 to 250 is a good guess. I said around 175 to 250 for library 1, and then around 150 to about 275 for library 2. So yeah, that's about right, Hannah-Lee. Okay, so a little bit more about these adapter dimers, because they're so important to make sure you get rid of them. So I really wanted to kind of hone in on this point about how bad these things are. So once we assess our library quality, of course, as I mentioned, we're looking for adapter dimers at fragment size. And this is what the adapter dimer looks like. I know I kind of described it in words, even though I had a picture here. So we see this P5, and then along with the priming region on the P5 side, and then we see our P7 side here with the index, and then the respective P7 priming side here. And so those have ligated to each other, which is causing our primer dimers, or adapter dimers, I should say, because primer is just one region of it. Adapter is the whole thing. So this library represents 0 adapter dimers, which is the dream. Here is just 5% adapter dimer, and these are calculated just by looking at the area under the curve. So even though this is a high peak, of course, when you're looking at the percent, you're looking at that area under the curve. And so what happens, Felix, to answer your question a little more thoroughly, so if we have, so in our control sample here, where we have 0% adapter dimer, or roughly 0% adapter dimer, the number of adapter dimer reads is 0.24% of here. PF stands for pass filter. So there's a filtering system, so that way you know how many of your sequence, how many of your reads pass filter, what percentage of them. And so in this run, we had about 69% pass filter, and that would be our sequence of our region of interest. And then we had about 0.24% of those reads were identified as adapter dimers. And this again is related to the 0% adapter dimer as indicated by the electropherogram for it. Now, if we have as little as 1% adapter dimer, it will now take up over 6% of the reads, which not too bad, not ideal, but still not too bad. But now if you just go up to 5%, so if you only have 5% of adapter dimer in your library, that will now take up 60% of your reads. So with just a 5% adapter dimer contamination, you are throwing away 60% of your sequences. So that is how important cleaning up these adapter dimers are. Now, if you go up to just 10% adapter dimer contamination, you're looking at almost 85% of your reads. So you might as well just throw away that sequencing run because you gave it all to adapter dimers. So this again is my, go ahead, Felix.
Oops, sorry, I was muted.
Okay.
So just to clarify, the reason for this is that even though it's, say, only 5% of the total mass, it takes up 60% of the flow cell?
Right. So remember, when we are loading on so essentially we create these libraries for all of our samples, and then we pool all of these libraries together. So we combine all of these libraries from all of our samples in both our control and our experimental groups, because remember, we have little indices on them, so we can identify where they came from at the end. And so we bring them all together into this pool, which is a certain concentration. And of course, that concentration contains, way more transcripts than what's actually going to be sequenced on the flow cell. And so when we draw up in our pipette from that library pool and put it on the flow cell, the smaller the fragments are that are introduced to the flow cell, they tend to latch on and to their complement really quickly, right? Because it's just a small region, this P5 or this P7, that's going to be complementary to the oligos that are attached to the flow cell. So if this is really small right here, so you need a smaller percentage of this that's complementary to the oligo, so it's going to find it and latch on, right? It doesn't have this like kind of long insert here that'll cause it to kind of flow through and might not have a strong bond there off the bat. So the smaller these fragments are that are introduced to the flow cell, the more likely they'll be to bind to a complementary oligo, and they'll do it quicker. And so as soon as they bind to a complementary oligo, that oligo is taken up now, so nothing else can attach there. And so what happens is that when you inject your sample into your flow cell, those small fragments are going to kind of latch on real quick, so much so that even 5% contamination will take up a lot of the spots on your flow cell for sequencing. Good question. And this was actually it was like a shock to Illumina too when this happened. And so they really harnessed in their training, which they have great training for library preparation. We actually have a POC that we kind of have on call. Anyone who has a NovaSeq 6000, which is the largest sequencer that Illumina offers, we kind of get tech support basically on call, which is really nice. And so this is one thing they really harped on in the training of making libraries. Okay, so now we have our libraries. We have sufficiently ensured that we cleaned up and gotten rid of any adapter dimers, and we can now start sequencing. So I'm going to walk you through what sequencing looks like on the Illumina flow cell, and then we're going to watch an awesome video that's going to explain it way better than I am. Okay, so in this little cartoon here that I pulled, any pictures or images that I use, I have references to them on the slides. So when I send out these slides, you guys will have all that information, and I encourage you to look and read up more about this stuff, because it takes a while to kind of wrap your head around. Okay, so we have our libraries, which essentially look like our region of interest with our adapters on either end. This cartoon also shows those adapters being ligated, so we're already past that step. So let's focus here. So now when we introduce our sample pools containing our libraries into the flow cell, what happens is those P5 or P7 regions are going to be complementary to the regions, to the oligos that are on the flow cell. And those oligos are actually covalently bonded to the surface of the flow cell. So of course, this bonding here is hydrogen bonding, which is not as strong as covalent bonding. So those little oligos won't move. And then so this hydrogen bonding happens to keep that there. And then once that hydrogen bonding happens, what occurs is yet another PCR step. This is how useful PCR is and why it was received the Nobel Prize, and it's one of my favorite inventions. So then PCR happens, and it basically makes a copy of that library strand. And so now we have a copy of our target sequence that is covalently linked to this flow cell, right? So it's not going anywhere. And once it makes that copy, it will wash away this hydrogen bonded initial library that we added. And so now we'll be left with basically the complement of the libraries that we added that are covalently linked to our flow cell. And then what happens is something called cluster formation. And so let's say that this pink here is P7, so then we have the P5 region here. So that will kind of bend over. And again, there's fluid flowing through this. So these are, wobbling around, and eventually it'll find its way down. And once it is, it'll hydrogen bond to its complement here, which again is also biotinylated to the, or covalently linked to the flow cell. And so that's gonna bend over, and then what's gonna happen is that a copy is gonna be made. So yet another PCR happens, and this is called bridge PCR, because when these flop over and attach with their complement, now this amplification is happening in a bridge formation. And so basically, this will then make a copy of that, and then it'll be denatured. And so you'll have two copies now, each of which are attached on to the flow cell. And then those will both fold over, and then you'll have bridge amplification again, and then that'll happen again, and then again, and again, and tens of thousands of times for each cluster until you have a ton of copies in a cluster. And so this is what it looks like in Illumina's reference guide. So again, we have our adapters now in purple and blue. And we have our region of interest depicted here in green. And so this, again, is just showing that bridge amplification. So this will fold over. It'll undergo a PCR step to create a copy. And then that will keep happening over and over and over again. And then eventually you're left with these clusters of all the same insert. Right, so this cluster here is going to have a different insert, a different green region than this cluster here, which is going to have a different green region than this cluster here, etc. And so those are all what are eventually going to become all of your reads are going to be generated from these little clusters. And then this is, now that we've generated all these little clusters, we are ready for sequencing. But I guess before we do that, any idea why we're making so many copies of the same thing to sequence? I think Dr. Heller might have mentioned this in his lecture, kind of one of the caveats of pyrosequencing or sequencing by synthesis.
To make sure the sequence is accurate.
Absolutely. So pyrosequencing or sequencing by synthesis is inherently fairly error prone. And to account for that, what happens during this bridge amplification is that there's tens of thousands of copies being made. So as this is being sequenced, each base pair is being sequenced tens of thousands of times within each cluster. So not only does it amplify the signal, but if it so happens that like 1,000 of the, 50, 100,000 sequences have an error in it, it'll be overwhelmed by the correct base pair. And so it kind of helps account for that error proneness of sequencing by synthesis by just making a ton of copies. Okay, so now we are ready for sequencing. So you can sequence either single end or paired end. The GeneLab standard is to do paired end. Paired end will, of course, give you more information, so I'm always a fan of more data rather than less. However, depending on cost and how much you have in your budget, you may opt to do single end sequencing. And so single or paired end sequencing, the first sequence always happens the same way. So what it starts off with is you'll have your, go ahead, Christina. I think you might be muted if you're trying to talk. Did you have a question? No question? Okay, if it comes back to you or if I don't address it, feel free to turn off your mute and shout at me. So as we're sequencing, we sequence the read one first. So read one we introduce, of course, primers that are complementary to that priming region of our adapter on the end that is not latched on first. So I think it's the P7 end. Yeah, so P7 I think is down here and P5, yeah. So we see our index here. That tells me that this is P7. Over here is P5. And so what happens is the primer that's associated with our P5 region, that complementary primer will be introduced. And so will, of course, polymerase as well as all of our labeled ATCs and Gs. So all of our labeled nucleotides. And so that will be primed here. And then we have polymerase that will latch on and start pulling in those nucleotides. And each, I know we kind of use cycle in a different context in PCR, but each base pair incorporation is considered a cycle in Illumina sequencing world. And so the number of cycles will, of course, dictate your sequencing length, or ultimately your read length. And so what happens is that you add this primer that's complementary to the priming region of the P5 side of your template, and then your polymerase will go on and it will incorporate the complementary base pair and give off the signal that is the color that represents that base pair, and a picture is going to be taken after each cycle. And after each cycle, the reaction is quenched and a new cycle begins. And so you'll go on and you will do your sequencing by synthesis, so taking pictures after each cycle until the setting that you set for the number of cycles is achieved. So question, if you have an insert that is 100 base pairs, so your region of interest is 100 base pairs, but you set your sequencing to be 150 base pairs, what happens? Anybody know? No guesses? So what happens is if your insert is 100 base pairs, but you set the number of cycles or your read length to be 150, this will continue. The cycles will continue for those 150. So when you have your sequence at the end of the day, it'll have your 100 base pairs of your region of interest, but then it'll also sequence into the adapter. So part of your sequence will be the adapter. So it'll continue to sequence into the adapter. Something to keep in mind when we start looking at the data later. Yeah, and so this will make up read 1. And then what happens is we want to read the primer, or read the index, right? And so then we have an indexing read. So this is showing, again, the same thing that's depicted here. So we have our I5 region, the priming on the I5 region happening, and then we have our sequencing by synthesis, each one, again, of these base pairs attached to a fluorophore. So as these cycles go on, it gives off a signal that represents each one of these base pairs, and that carries on and gives you your forward read. And then after your forward read, the forward strand reagents are washed. And actually, I lied here because what happens after this is sequencing of your I7 primer. And so after read 1 happens here, those reagents are washed away, and then you have the primer for your I7 come on, and it's actually going to continue to sequence, right? So we always sequence in the 5' to 3' direction. So this is going to continue to sequence in this direction, and that's how you'll get the sequence of your index, which is what you need. So you have your read 1 sequence happening, and then you have all those reagents washed away, and then you have your primers being introduced to that I7 end, and that will allow sequencing of your index. So your first read is going to be your forward read. Your next read is going to be your P7 index read. And then after that happens, then it washes away, and we actually have these things fold over, complements made, and this kind of gets reversed, and we have read 2 sequencing happening. So what happens is this is all washed away, this folds over, we make a copy, and so now we have the part of I5 over here that's attached to the flow cell, and now we sequence in that direction, or in the read 2 direction. And then once that read 2, the number of cycles that you have for the length is completed, those reagents are washed away, and you do your sequencing of your I5 index if you have an I5 index, which I know it's not shown here, but we do. Questions about sequencing, cluster formation or sequencing by synthesis before we watch one of my favorite Illumina videos? No questions? Okay, great. If this was all kind of a lot of information and too much, hopefully the sequencing video will bring it all together. But before that, I just want to go over the sequencing parameters that we use. So for our read length, the longer read increases our gene identification confidence. Anybody have any guesses as to why that is? So why would a longer sequence increase your confidence about where that sequence came from? Shorter sequences can be repeated in other genes. Yes, less puzzle pieces to put together. Also a good answer. Smaller reads might be shared between different sections. Greater specificity, so not as random. Yes, all absolutely right. So of course, our genomes have a lot of repeats in it. So there are segments of transcripts that have similar, if not the same sequence. And so the longer your sequence is, when you go to try to identify where it came from, the longer it is, the more likely you'll be able to map it uniquely to a region on the genome, and therefore you'll be more confident in your call of where that came from. And so we really want to get the longer reads, the better, but of course we want to maintain high quality. And so one of the caveats of sequencing by synthesis is kind of like the polymerase gets tired, just because we really haven't optimized the conditions quite as beautiful as they are inside of our cells to allow for the polymerase to kind of sequence forever. And so the optimization, like I said, Illumina longest reads are about 500 cycles total, so you could do paired end 250. We do more common paired end 150 is our standard. So GL just stands for GeneLab. Oh, and Marie just joined. If anyone has any questions about library prep, she can answer them. Sequencing depth. So what is sequencing depth? Anybody know? Anybody know what sequencing depth means? How many times a piece is sequenced? Close, Felix. So it's how many sequences are generated for each sample. So how many times each piece, I took that to mean like each insert is sequenced. So each of your inserts, once it attaches onto a flow cell, should be sequenced roughly the same number of times, right? Because we create those clusters that have tens of thousands of copies. So each piece is going to be sequenced roughly the same number of times, but how many pieces or how many inserts or fragments of RNA that you sequence per sample is going to be your read depth. So the more sequences you have, and I know I've been using the word sequences and reads kind of interchangeably because they are. So reads are just short sequences. And so the number of short sequences that you have per sample is going to be your read depth. And so if you have greater read depth, you have more sequences that you have accessible for that sample. So you will increase the likelihood of detecting low abundant transcripts. So if you have transcripts that aren't very highly expressed, and you're really interested in those kind of lower expressing transcripts, you want to sequence deeper, because the more inserts that you sequence, the more likely you'll be to pick up those low abundant genes. It'll also allow for you to detect novel transcripts, and it helps you to quantify isoforms, the more sequencing reads that you have. And probably should ask this as a question, but greater sequencing depth is necessary for Ribo depleted sample as opposed to poly A enriched samples. Any idea why that is? Any guesses of why we would need a greater read depth when we prepare our libraries with the Ribo depletion method compared with the poly A selection method? Because there's more variety in the ribo-depleted samples, yes. To make sure you don't lose a targeted mRNA, yes. Absolutely right, Eowyn and Nicholas, great job. So the greater depth you sequence, you're going to pick up more stuff. And remember, when we have ribo-depleted samples, not only do we have messenger RNA, but we have other types of RNA too, such as long non-coding RNA, microRNAs, etc. And so especially if we want to do differential expression analysis, which is specifically looking at expressed genes or messenger RNA, we really want to make sure we cover as much of it as we can. And so we want to sequence a little bit deeper in our ribo-depleted samples as compared to poly-A, which have already been enriched for that messenger RNA, like we saw a few slides ago. And our GeneLab standard for mammals prepared with Ribo Depletion is 40 to 60 million reads per sample. So that's our target goal when Marie and the rest of the sample processing lab is calculating how much RNA or how much library from each sample they want to add to a pool for sequencing. They do their calculations to try to get as close to this 40 to 60 million reads per sample as they can. And just a side note here that if you have, of course, a certain budget and you're doing a sequencing experiment and you have the option of sequencing an extra 5-10 million reads per sample or adding on more biological replicates, always prefer more biological replicates. What do you guys think is better? I think I kind of covered this a little bit, but paired end or single end sequencing, so sequencing in one direction versus sequencing in two directions. Paired end, yeah, Hannah Lee. We want to see paired end, we get more data out of it, and then we also have that nice option of overlap, and that helps with isoform identification and things like that. So we prefer paired end, and that's part of the GeneLab standard as well. Okay, so now you guys are going to watch one of my favorite Illumina videos, and I have the link here, and I'm actually going to post this link. Oops. Let me escape, just because I'm going to post this link in the chat so you guys can watch this a million times. Maybe that's an exaggeration. I've seen this video over 20 times for sure. So I'll go ahead and play it now. It's about 5 minutes long, so not too bad. And it's going to go over the sequencing by synthesis that I just talked about.
The Illumina sequencing workflow.
I just want to pause. Can everyone hear it? Let me know. Yeah, okay, cool.
Is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of 2 types of oligos. Hybridization is enabled by the first of the 2 types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in 2 single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescent intact nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized, and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in BaseSpace Sequence Hub. Discover the possibilities of next-generation sequencing.
So how do you guys like that video? It's one of my favorites. Like I said, I've watched it definitely more than 20 times. It takes a couple of watches before you grasp what's going on. So again, I put that video link in the chat. So watch it as many times as you need to until you're comfortable and understand the sequencing by synthesis process. It really is just such a useful video. Is the U addition in clustering or sequencing? So the U addition actually happens in library preparation. Is that what you're talking about when we create the strandedness, Rashi? Yeah, okay. Let me go back to that actually. So in the library preparation step here, first, after we remove our ribosomal RNA, chop up our remaining RNA of interest, then we start to first create the first strand of cDNA, and that's created with Ts. So this is the reverse transcriptase process that we saw in that RT-qPCR that we saw earlier. And so that first strand of cDNA contains Ts. And then when you make the second strand of cDNA, so basically after this, there's an RNase-H step that I didn't mention that actually gets rid of the template RNA. So that way you're just left with this first strand cDNA. And then that goes through another synthesis to create the second strand of the cDNA molecule. And then that's where those U's are incorporated. And so we're here at this part when we have our first strand, which has T's, and then we have the second strand, cDNA, which is complementary to that first strand, which has U's. And then we go through and add on the polyadenylation to our 3' ends, which allows us to ligate our adapters to create our initial library product here. And then we go through the step of amplification. And during this amplification step, the polymerase that's used doesn't like U's. And so as soon as it runs into a U, it'll just stop making a copy. And so kind of in this final amplification step, which is where we make a copy of our complete libraries, it'll only make a copy of the library of that first strand of DNA. And so that way each strand is in the same direction or same orientation. And that allows for the strandedness of the library. And so then coming off of this step, we do another filtering step to get rid of the polymerase, to get rid of the free nucleotides that are there. And then we also remove the template here with the U's. And so we're just left with stranded libraries. And so those stranded libraries contain T's, and then that's what's ultimately put on the sequencer and sequenced. Make sense? Makes more sense. Great. Okay. Felix said what? What do you need to go over again? I'm happy to. What's the advantage of strandedness? Good question. So as we're going ahead and, I guess I'm gonna go here first. So as we're making this first strand synthesis, actually, no, I don't have this straight in my head right now. I used to know that answer, but I don't want to confuse you. So let me get that straight in my head again. And then tomorrow morning, I will talk to you first about why strandedness is important. Unless, Marie, do you know off the top of your head right now?
I read the theory of why strandedness. So originally when they developed RNA-seq, it used to be not stranded, right? But then for some types of experiments of interest, so let's say, for example, you're looking for a virus or whatever, and you want to know the origin, the template that it came from. So things like that, you would want to know the strand that it was originated from when you do the synthesis from, right? So that's the advantage. So you want to know the origin of your RNA source or your RNA input, if that is something of interest to you in your final work.
That makes sense. That sounds right. So you're always looking at, and when you're doing your aligning, rather than having to align to the genome in two different directions, you only have to align in one direction, and that also increases accuracy of your counting.
Yes. So yes, but originally it was not thought about.
Yeah.
As the direction of the source of your RNA.
Originally, yeah, it wasn't. And so you had some reads aligned in one direction, some reads aligned in other directions. And then when people were quantitating, some people were quantitating reads only aligned in a certain direction. Other people were quantitating reads aligned in a different direction. And other people were quantitating reads no matter what direction it was aligned to relative to the reference genome. And so this is another way to kind of standardize things to reduce the variability when you repeat experiments. So I hope that makes a little bit of sense, but I'll also look it up too, again, and make sure that how we're thinking about this is right. Got cut off in our responses, Felix?
I think he means that when he was originally typing, he typed what? And then he typed his full question.
Oh.
what Lauren said.
Gotcha.
See, another question. So when you're doing RNA-seq and you have an mRNA, how do you know if there are genes on both sides, how do you know which of the gene generated the mRNA?
Well, that's another way to know from looking at strandedness. Right? So if you have your mRNAs, right? And like you said, those mRNAs could be derived from either strand. So from the forward template strand or for the reverse template strand, right? If when you generate your libraries, you're only making your libraries in one direction, when you go back and align it to your reference genome, you'll know because you only mimicked the reverse complement of the original strand. And so you'll be able to determine which strand that transcript came from. Whereas if your first strand and your second strand were both amplified, then you wouldn't be able to go back and know which strand was the template for the mRNA. I don't know if that makes sense. I'll make some pictures.
Yeah, I think I'll probably need pictures and maybe just to think about it earlier during the day. Yeah.
I'll make some pictures and we'll start
Thank you.
with a little more discussion about strandedness and identifying the origin of the mRNA. Good question. Okay, so we have officially made it through sequencing. Any other questions about sequencing? No more questions about sequencing? I encourage you guys to watch that video a few more times. I'll find some cartoons or make some myself to kind of address Felix's question about strandedness. Yeah, so I know a couple of people have been kind of getting cut off a VPN here and there from the lecture series. So I'll make sure tonight that I condense these videos and put them in a Google Drive. So today's lecture, at least, I'll make sure that I get those up on a Google Drive and send you that link, hopefully within an hour or two after we end for today. And so that way you guys can have that, and I'll also send you the slides. So that way you guys can all have that tonight. Should get that about 2 hours after we end for the day. And so that way you guys can have another look at it tonight before bed or first thing tomorrow before we start. Yeah, it's definitely a lot to absorb for sure. That's why we called this a boot camp. Want to push your brains to the max. Get the info. Oh, yeah. So Winnie and Kristen sent the information with their with the opportunities that they had presented to me already. So I'll get that up there tonight as well. So I do want to get through the RNA-seq workflow lecture until the end of looking at the quality of trim data. So that way tomorrow we can start with a quick review and some information about strandedness, and then hop right into the start of the Jupyter notebooks. So I do have about another 20 to 25 minutes of lecture that I want to get through before the end of the day, but I know that I just gave you guys a lot. So do you guys want to take a quick 5-minute break before we get into the processing side of things? Yay, nay, yes, I'm seeing thumbs up. Okay, Phil, you had your hand raised?
Yeah, I just wanted to make a quick point. So we've discussed two technologies today, microarrays and Illumina. And at some point in the next 10 years, probably they'll both be made obsolete by nanopore sequencing. But that doesn't mean that what we're doing today isn't valuable. It's going to be extremely valuable for a long time because there is so many, there's just such a huge volume of data that has already been collected by these two technologies that's just waiting to be either looked at for the first time or looked at for the second time. So, for a long time after everybody's doing nanopore, this knowledge that you're learning today is going to be super valuable.
Yeah, that's a very good point. The cool thing about sequencing data is, even unlike microarray data, which is, just looking at relative expression between two samples, whereas the sequencing data is the sequencing data. And so that's kind of immortal. So you can go back and revisit this data as much as you want, look at it in as many different ways as you want, come up with new questions, new hypotheses as we get better and better builds and more accurate builds of our reference genome. These sequencing data can be realigned again and again and again to get more and more information out of the same sequencing data. So that's one of the things that's really cool. And also one of the purposes of GeneLab is to reuse data and come up with new hypotheses and new conclusions about what's happening specifically in the space environment, but really that's relevant in any context. So I'm going to give you guys that 5-minute break. If you guys can come back here at 4:35, and we will get through the next 10 or slides or so before we call it a day. So everyone come back at 4:35 p.m. and we'll pick up where we left off. was muted. Welcome back, everyone. Hopefully you guys had a chance to grab some water, a little stretch, maybe even step outside. I know I just gave you a ton of information in that last segment there. Don't call this bootcamp for nothing. So we are gonna just continue right on up until assessing the quality of trimmed sequencing data. So I am currently sharing my screen. I never stopped recording since it was just a 5-minute break. But I do want to make sure, are you guys all able to see my screen?
Yes.
Awesome. And then Lauren, you're still a co-host, right?
Yes, I am.
Great. Okay. Congratulations. We have officially sequenced some data in theory. And now we're ready to move on to data processing. So this is why we're all here. I know it came with a whole lot of background information, but as dense as that information was, it is, and I hope you'll see the usefulness of kind of knowing the whole process from start to finish as we're starting to analyze these data. So this is just a general overview of the RNA sequencing workflow. And so we're going to start here with raw reads or raw sequence data. And the first thing we're going to do is take a look at the quality of those data. And so we're going to do this with a program called FastQC, and then we're going to compile the FastQC reports from all of our samples into one nice set of graphs called MultiQC. And there's actually a lot of information in that quality data, so that's what the bulk of the rest of the time today is going to be spent on, is just taking a look at how to assess those FastQC reports. After we assess the quality of our raw reads, we will then know if we should trim or filter our raw data before we move on to the alignment step. So we'll do that next, and then after we trim or filter our raw data, and I'll get into more details in those slides, we're going to again check the quality of the data to see if we did actually improve the data with trimming and filtering. After we trim or filter, we're then going to be aligning those trimmed data to our reference genome. So this is where we try to figure out where all of those sequences were derived from so that we know which genes are actually being turned on or turned off in our spaceflight samples compared to our ground control. And in order to do that, we have to create an index. So this is going to be unique to the tool that you're going to be using to do that alignment back to the reference genome. So we'll get into that starting tomorrow. And once we align it, we assess how well our alignment went. And again, we use MultiQC to do that. And then after we align our reads back to our reference genome, now we know where they came from. So now it's time to count them. So then we start counting how many reads are associated with which transcript. And we do that using a tool called RSEM. And RSEM, just like the tool that we use to align called STAR, requires its own version of the genomic index. So we have to do that first. And then we quantitate those reads. And then after we count them for each sample, we combine them all together into a counts table, which is why Lauren spent so much time showing you guys how to mess around with data tables in her R intro notebook. And then after we combine them all together, we have to go through a normalization step to account for any differences in the samples that were caused due to technical discrepancies. And then after we normalize our data, we can then go on to perform differential gene expression and identify which genes are up or down regulated in our spaceflight sample compared to our ground control. So that's how the processing is going to happen in summary. So let's dive in and get started with this first step, which is checking the quality of our raw reads. Before we check the quality of our raw sequence data, let's take a look at what it looks like. So raw data generated from the sequencer are stored in FASTQ files or base calls. If they are stored as base calls, those are then converted to FASTQ files. So for the purposes of this bootcamp, we're just going to start with the FASTQ files. So what is a FASTQ file? A FASTQ file contains multiple reads, and each read is formatted in this very consistent structure every time. So what you notice is each read consists of 4 lines. And so line 1 is always going to begin with an @ symbol. And then it's going to be followed by this information here that looks like it's a code. It kind of is. It's information about the sequencing run, such as the sequencing platform that was used, the run number, the flow cell ID. So I should mention that so the flow cells that we use that have the lawn of the oligonucleotides where the sequencing happens, we have IDs for those. We also know the location of all the dots on those flow cells. And so that cluster location is another thing that's kind of indicated in this first part of this first line. And then that's followed by the read number, forward or reverse. So here we see the number 1. Any guesses if 1 indicates forward or reverse? Forward, yeah, Diksha, you're absolutely right. So 1 indicates forward. Reverse is usually indicated by a 2. That can get complicated for those of you that are going to be doing an internship and choose to do the UMI. You'll kind of see how that could be different. But yeah, in general, 1 indicates forward read and 2 will indicate a reverse read. So this is indeed, we're looking at a forward read. And then finally, we'll see the sample index. So that is indicated by this 6 base pairs here. So in this sequencing run, the index that was added to this sample is 6 base pairs. So basically any read that had these specific 6 base pairs were identified to the same sample. And then just to give you guys a heads up, although almost all read headers will at least have the forward reverse info as well as the sample index, unfortunately, some databases will remove kind of all of this fairly useful information about the sequencing run itself, which always frustrates me. At GeneLab, we don't. And so if you guys ever see a read header where that first line is missing this information and something else is in place of it, it's because the database or whoever's storing that data changed it. Line 2 should look very familiar to you guys. So as you can see here, it's a bunch of A, T, C's, and G's. So these are our base calls. And the number of base calls should be equal to the number of cycles we set our sequencing for, which is ultimately our sequence length. And so I think this one here is maybe 100, it looks like. And then, so that's always going to be in the second line of each read. The fourth line, or the third line of each read is just a separator line. So for Illumina sequencing runs, it begins with a plus sign. That's really is the most common convention. But I've also seen reads that don't have any symbol here on this third line. It's just a completely blank line. I've also seen reads where it's a symbol other than the plus sign, but it's almost always a plus sign. And then the fourth and final line are quality scores of each base call. And these are written in a code. For red 33 encoded ASCII characters are used to represent the quality of each base. So it basically tells us how confident it is that when it calls this base as a G, that it actually is a G. And just to take a look a little bit more at what those ASCII characters represent, you can kind of look at the ASCII base. I think I have a link to this. If not, there's definitely a link in the Jupyter Notebook to this table. So the ASCII base 33, this is for these different instruments. Illumina, again, is the most common and the one that we use at GeneLab. And just to kind of show you what these ASCII scores represent. So the ASCII symbols are all the way on the right column. And so that's what you were seeing in that fourth line. And so they each represent a probability of an incorrect base call. So just as an example, if we take a look here at the ASCII character represented by a question mark, that corresponds to a quality score or a Q score of 30. And a Q score of 30 means that there is a 1 in 1,000 chance that that base call was not right. And so do you remember when those clusters have, how each cluster has tens of thousands of the same read? That's what it means, is how many for that cycle was that called a G in each one of those tens of thousands of reads in each cluster. That's how it determines these Q scores. And so a Q30 is obviously really good because that means an error rate or an accuracy rate of 99.9%. So if you have a Q30, again, represented by a question mark, you'll have an accuracy of 99.9%, which is awesome. And if you recall from the previous slide, we had a lot of J's and K's in there. So really high quality. which is pretty consistent with coming off the Illumina sequencer, as long as you did your library prep correctly. So, of course, it would be pretty cumbersome to have to go through and look at these ASCII scores for every single base pair, for every single read, especially when you're talking about, we said 40 to 60 million reads per sample. So a program called FastQC was created to assess the quality of sequence data and then provide a nice QC report that will allow us to quickly evaluate the quality of our data. So I'm going to walk you through the data that's given in these FastQC reports. So if you want me to explain something else or explain something in a different way, or if you're not fully understanding something, either unmute your mic and ask me to explain it better, or shoot a message in the chat. So one of the first pieces of information it gives us is just basic statistics. So we can gain some general information just from looking at this basic statistics. So the first question I'm going to ask are these reads forward or reverse reads? You can either type in the chat or shout it out loud. Any guesses? Guessed reverse. Zinru, why did you guess reverse? You're right. I saw R2 in the file name, right. Okay, so yeah, that's exactly right. So in this file name, I have an R2 here, R standing for read, 2 standing for the second of 2 reads, or the reverse read. So informative bioinformaticians will always label their FASTQ files with an R1 or an R2, or at least a 1 or a 2 to indicate forward or reverse. Unfortunately, not everyone does. So hopefully you'll see it in the file name as we are training you guys to be the best bioinformaticians that you can. Make sure that anytime you guys are working with FASTQ files, if you are responsible for naming them, make sure the forward or reverse is indicated in the file name. What's the read depth for this sample? Any guesses on that? No guesses on the read depth? Yeah, Zinru again, absolutely. It's totally right. It is 100,600,211 reads. So just a quick question. You don't have to tell me the exact number. Just tell me the what mathematical operation you would perform on that number to know how many lines the FASTQ file would have. So if it has 100,600,211 reads, what mathematical operation would you have to perform to that number to know how many lines are actually in that FASTQ file? Any guesses? How many lines are in each read? 4, yeah, multiply by 4, absolutely right. So basically, if you were to open up that FASTQ file with a cat command and pipe it into another command that we didn't go over to do a line count, you would see over 400 million reads or over 400 million lines in that file, for every 4 lines representing 1 read. What's the sample read length? 150, yes. This one's a little bit more straightforward. It tells you sequence length 150. So that is the number of cycles or the read length that we had. I guessed 100, I was wrong. Prematurely. And then the other thing that it shows here is percent GC content. We'll see that again in another graphical representation too. So that's the information just from the basic statistics. So now we're going to get into a little more of the quality. So if you remember, we were just looking at the ASCII characters, which represents a quality score or a Q score. And that is done for each base that's called, right? So if you look here, this is showing the per base sequence quality. And as you can see on the x-axis, you see the position in the read. And so, of course, there's 150 positions. Why is that? The length of the read. Yeah, Hanali Rashi, yes. Length of the read. And so because of the length of the read, the quality is assessed at each one of those positions, and we're giving kind of a box plot here to show the basic, the average, as well as kind of the outliers for each read in this sample. And so it's pretty nice because FastQC gives you an indication with green, yellow, red of the quality. And so I have a couple of questions based on this. So over here on the y-axis, if I didn't make that clear, that's our Q score. So that's our quality score. So it's basically saying here that everything above a Q score of 20, which is 99% accuracy, they're considering good quality, and everything above 28, which is pretty close to 99.9% accuracy, is really good quality. So a couple of questions. Do you notice anything about the quality as you move towards the end of the read? Why are some columns combined? Good question. I think it's just to fit on the screen. So if I were to have stretched out the screen here, when I was looking at this, it probably would have give you high resolution. Yeah, it deteriorates, it gets worse. Yeah. So as you move towards the end of the read, the quality kind of drops off. So any idea why that is? Any guesses? Watches from Illumina. So yeah, that's true. After each base is incorporated, there is a quenching that happens. So that's one hypothesis. Another hypothesis is that perhaps the polymerase kind of getting tired and falls off a little bit towards the end there. And then let's see, these data are for the reverse reads. Do you think the quality would be different for the forward reads? Any guesses? I think it would look kind of similar. Yeah, Hannah-Lee, you would think, right? But in actuality, the quality of the forward reads tends to be better than the reverse, and we're not entirely sure why. I mean, it's true that the sequencing of the forward read happens first, is the very first read, but it's all the same technology. It uses the same reagents. You know, it does the washing to clear out anything that was there before going on to do the reverse read. So it's still kind of poorly understood as to why the quality of the reverse read tends to be a little bit weaker than the quality of the forward read. Not much, but a little bit. And that's fairly consistent. And yeah, still not horribly sure why that is. Okay, so moving on to another way to look at the quality data here. This is again looking at the quality scores, but it's looking at the per sequence quality score. So what this kind of does is now we have our Q scores on the x-axis, and on the y-axis, we have number of reads. So this is basically just giving you an overall summary of how many reads have what level of quality. So as you can see here, we have the majority of the reads, right, have this really high quality. And so would you guys consider these data to have good quality overall? Yeah, for sure. So another thing that we get to see, as I kind of mentioned at the basic statistics, is this actually that's the next one, sorry, is the per base sequence content. So this is looking at the sequence content across all bases, and you're really looking, I guess let's do the bullet points. So these values really should be fairly consistent across the read length in an unbiased library. Any idea what I mean by unbiased? Any guesses? Not favored towards, yeah, pretty much, Awen. So not favored towards anything in particular. So there's no contamination, and basically there's high diversity amongst the calls, meaning that for all the reads for each cycle, we don't have like an overwhelming amount of A's being called in the same position. or an overwhelming amount of T's being called in the same position. We want to see that each call is fairly balanced, and that overall we have roughly the same level of ATCs and Gs. So if the library is biased, what you would see is spikes in the data. Although we do see some spikes here in the beginning, that's actually has to do with the nature of the RNA-seq primers, and they just tend to cause some wobble at the beginning of the reads. So this is a pretty common thing that you'll see with the per base sequence content. Is a little wobble here at the beginning for RNA sequence data, and then it kind of levels out again if your library is good quality. Okay, this is a GC content graph, and so what this does here is it shows you what we expect the GC content to be. So we expect GC content over the reads to be normally distributed. We have number of reads here on the left, and we have the mean percent GC content on the x-axis. And the actual GC count per read is in the red line. And so is the GC content normally distributed across all reads? Ish. Yeah, kind of. Yeah. It's pretty much where I'm at too, kind of here. So what do you think the peaks outside of the normal distribution indicate? Any ideas? What the peaks outside of that normal distribution could tell us about the data? Higher or lower than normal, areas of higher GC content. Yeah, what do you think that means? If we have kind of these spikes in GC content that happen, what could that indicate? What could that tell us about the nature of the sequence or where the sequence came from? Okay, so then just have to tell you guys, methylation, good guess. We're actually working with RNA though, so I mean, although you are right, so there is epitranscriptomics, but you would have to specifically be looking for that methylation. So there is a field called epitranscriptomics, and we actually do have some data sets that have epitranscriptomic data, and so that would be how you would look at methylation. So this type of sequencing doesn't really show that. But those kind of weird peaks outside of the normal distribution probably indicate some form of contamination. So maybe we have some adapter dimers in here, or maybe we have, we actually spike it with phi-X to help with the diversity of the sequencing cycle. So maybe we have too much spike in of phi-X here. There could be a number of different reasons why we might have these peaks, but they usually indicate some form of contamination. It could also be some RNA or ribosomal RNA that was still in there. Okay, so looking at the next thing here, the per base N content. And I know I have 3 more slides that I want to get through fast QC, and then we can pick up the rest tomorrow. So I might go about 2 or 3 minutes over. So this per base N content here, this is looking at an N across all positions. So what happens with Illumina sequencing is that if it can't confidently call a base, then it'll put an N in there. And so, I just said that. So how often was a base unable to be called in these reads? Almost 0. Yeah, once maybe, yeah. So maybe just like a handful of times, very rarely here at the beginning of the read. And again, Illumina has just gotten so good with their technology that's pretty common. So the next thing we're going to look at is another way to see sequence length distribution. So this we kind of saw from the beginning, but we see here that the sequence lengths for everything is 150. So we have consistent sequence length for all samples. Length by read should be the same. I kind of answered this already. Yes, they're all the same. What is the read length for these data? I know you guys already answered that at 150 base pairs. So 2 more slides here. Next is adapter content. And so this, do you remember when we were talking about what happens if your insert or your sequence of interest is shorter than the number of cycles that you're using to sequence? So if your sequence of interest happens to be 100 base pairs, but you're sequencing at 150 cycles, so your read length is 150 cycles, it will sequence into the adapter. Exactly. And so this fast QC program actually checks for that. So it has a set of oligos that match the adapters that are used for common types of sequencing instruments. So you see a variety of different Illumina universal adapters and some other platforms, Nextera and Solid platforms it has on here. So as you can see, I guess let's, does this sample contain adapter sequences? Or does this does this sample contain adapter sequences? Yes. Yes, it does. Are these adapters, what kind of adapters are these? What type of universal adapter? Oh, so I guess there's only one label here. Yes, this is an Illumina universal adapter, which means that this was sequenced, or at least the library preparation was done with an Illumina kit. And so where are the adapters relative to the read position? Near the end. And again, any idea why? Why they're at the end? So they're near the end, since it's shorter and contains. So they're near the end because if you remember when we are sequencing the read, we kind of start. So if you kind of get the picture in your mind of, let me just show you. I have pictures. Let's use them. Okay. So as you're going on here. Zoom this in. So if we're taking a look at the sequencing, we're going to start here at this primer region, and we're going to start sequencing right into our region of interest. And so as you go on, this is going to be the 5' region of the sequence that we got. And so as you continue on, if you keep going, that's where you'll start sequencing into your adapter. So that's why you usually see, almost always see your adapters at the end of your sequences or at the 3' end, because of the nature of where the sequencing starts. So it starts at the start of your insert, and then if it goes forward, that's where you'll see the adapters in your data. Okay, guys, last slide for today, and then we'll call it a day. And I apologize, this is kind of a doozy of a slide. So the next thing we want to look at is sequence duplication levels. So I know we touched on duplication a little bit, and so I'm going to kind of dive into that a little bit more. So there are actually two general kinds of duplications that can happen. So one, which I haven't talked about yet, is actual biological duplication, right? So if you have a transcript that's highly expressed, so there's a lot of copies of that transcript, when we go through that first step of library preparation, so after we remove ribosomal RNA, and then we start chopping up the remaining RNA to get our short inserts that we're going to sequence, although using the metals, it's a divalent cation that's actually used to chop up that RNA. It's supposed to be unbiased, but there always is some slight biases. And so what can happen is that if you have multiple copies of a transcript, even though that cutting is supposed to happen randomly, it could cut a duplicate of a transcript in the same location, which will give you the same exact insert. And so that came from biology, right? That just happened to be multiple copies of the transcript from the start. So that's called a biological duplicate. And so those we're okay with, right? Because that actually came from the sample. It was derived by biology. It was nothing that we did. And then that other form of duplication is going to be a technical duplication. And actually, we're going to start tomorrow after we talk about strandedness. We're going to start tomorrow by looking at different types of technical duplication. But technical duplication is another way that you can get duplicated reads. And so one way to get that we already talked about, which is during that PCR step where we actually get duplicated reads. And so what this part of the FastQC does is it kind of gives us an idea of how many duplicates we have. And so this is used to determine how unique our reads are. The blue line indicates the percent of duplicated reads at specific duplication level. So our duplication level is on our x-axis. On the y-axis is percent of total reads. And so read duplication can either be biological or technical in origin. I guess I just kind of answered that for you. If you have a very diverse library where all reads are unique, 100% of your reads would have a duplication level of 1. However, it's common, specifically when working with enriched libraries, to have some level of duplication. Any guesses as to why that is? So we have a specific step in our library preparation that we kind of talked about that could introduce this. So it's that final PCR amplification step where some of those more abundant transcripts we could artificially introduce a duplicate doing that PCR step. So that's why it is common to have some level of duplication when doing RNA sequencing. And so lower duplication levels, so that means that the sequences that have anywhere from 2 to 9 copies, again, of the same exact sequence, that's what a duplication means, are likely reads derived from what we would consider interesting or genomic regions, so such as your mRNA. However, if you're looking at more moderate duplication levels, such as anywhere from around here, like the 10 copies to 100 copies, those are likely derived from some form of contamination, like ribosomal RNA that we didn't fully get rid of, or another highly repetitive genomic region. And so you can see in this sample, we have quite like 10% of the reads have 10 copies. We have another 10% of the reads that have 100 copies, about 5% of the reads that have 50 copies. So those are duplicated reads. And so the really high duplication levels, so over here, so the reads that are duplicated over 100 times or more are indicative of a library issue. So that usually indicates adapter dimers, or it indicates too much Phi-X, if you're spiking out Phi-X, or some other contamination. And so I don't want to alarm you guys too much. But I guess I first should ask, I know I kind of already gave this answer too, but are duplicates present in this sample? And should we be concerned? Any answers in the chat? No, it's late. Last question. Yeah, so yes and yes. Duplicates are present. Yes, we should be concerned, but I do want to give the caveat of this assessment. In order to preserve computational time, it actually only looks at the first 50 base pairs. So it's not looking at the full 150 base pairs of the reads. It also only looks at the first 200,000 reads. So we have over 100 million reads in here, and it's making this graph based just off the first 200,000. So there are those caveats to keep in mind, and it also doesn't distinguish between biological duplicates or technical duplicates. So that's another thing to kind of put us at ease. So although this does say that over almost 75% of our reads are duplicates, which means that we only have about 26% of unique reads. So that's what it's saying up here, percent of sequences remaining if you were to deduplicate is only about 26, which is alarming. You do have to take into account the caveats of this analysis. And so again, that trying to make this more accurate is something that some of you guys will be working on this summer. Okay, I know I'm 8 minutes over, so I am going to go ahead and stop here. I am going to upload a copy of the videos of today's lecture part to a Google Drive. I'm probably not going to do too much editing just so I can get it up there quickly. And I'll shoot you guys all an email with the link to the Google Drive right after I get it up there. So thank you guys for hanging out with me a few minutes over. Anybody have any burning questions before we end for the day? This was a hard slide. Can we repeat it? Absolutely, Felix, for sure. Yeah, it was a hard slide. I did want to put it in your guys' head so that way when you sleep on it, hopefully some of the information will get transferred into your long-term memory. But yeah, we'll definitely start first by going over the issues with the template orientation, so the strandedness, and then we'll get into duplication. And then I have about 5 more slides before we'll start playing around with the Jupyter notebooks and start analyzing the data. So thank you guys all so much. And I will see you tomorrow morning. And if you guys have any questions for me, you guys can go ahead and write them down somewhere, and I'll be happy to start tomorrow by answering some of your questions from the dense amount of material we went over today.